Good morning, children. Today I am going to take the lesson Reproduction in Flowering Plants, Chapter 2. You all are aware of the process of reproduction, the significance of reproduction, and the types of reproduction. The types of reproduction. There are two types, sexual reproduction and asexual reproduction. In this chapter, we'll be dealing with the sexual reproduction in flowering plants. The first event in sexual reproduction is gametogenesis. What is gametogenesis? As the name suggests, it is the production of gametes from the gamete mother cells by the process of meiosis. So once again, what is gametogenesis? It is the production of gametes from the gamete mother cells by the process of meiosis. And I want you all to recollect the 11th standard cell division chapter, meiosis. How many daughter cells are produced as a result of meiosis? 4 daughter cells. So that holds relevance in this topic. 4 daughter cells are produced as a result of meiosis. And how many daughter cells are produced as a result of mitosis? 2. So remember this point right across the entire session. Now in plants, there are the two gametes, the female gamete and the male gamete. The female gamete is called the megaspore and the male gamete is called microspore. And so, the production of megaspores from the megaspore mother cells by meiosis will be called megasporogenesis. Similarly, the production of microspores from the microspore mother cells by meiosis will be called microsporogenesis. So, there are two gametogenesis, megasporogenesis and microsporogenesis. In today's section or slot, I'll be dealing with megasporogenesis. Also remember, this is a frequently asked essay question for the board exams. So your first point should be the definition of megasporogenesis. Once again, I repeat, what is megasporogenesis? The development of megaspores from the megaspore cells, mother cells by the process of meiosis. The second point should be a brief structure of the female reproductive organ, that is the carpel. What are the parts of a carpel? The three main parts are stigma, style, and ovary. Within the ovary lies the ovule. I am placing the ovule inside the ovary. And this ovule is connected to the ovarian wall by a stalk. I am highlighting the stalk by a stalk called funicle. So your second point should be a brief structure of the carpel. The carpel comprises of 3 parts, stigma, style, and ovary. Within the ovary, the ovule is located. Now, I am placing the ovule outside. The next paragraph should deal with the structure of an immature ovule. An immature ovule comprises of, it is connected to the ovarian wall by a stalk. As highlighted earlier, I have re-highlighted this stalk. It is the funicle. And this ovule is surrounded by one or two protective layers called the integuments. The integument, which is highlighted in pink color, encircles the ovule but leaving a gap at one end, and this gap is called the micropyle. So the ovule is enveloped by one or two protective layers called the integuments. Now enclosed within the integuments is a mass of cells. Can you see the blue color cells? A mass of cells called nucellus. Once again, the description of the ovule. The ovule is surrounded by one or two protective layers called the integument. And the integument encircles the ovule, not completely. It leaves a gap at the base, and this gap is called micropyle. Within the integument is a mass of cells called nucellus. Now, did you notice among the cells, among all the cells are uniform sized, but one cell which is at the micropylar end, I've highlighted in green color, is rich in cytoplasm with a prominent nucleus. This nuclear cell serves as the megaspore mother cell. So, one of the nuclear cells which is located at the micropylar end, which has dense cytoplasm and a prominent nucleus, serves as the megaspore mother cell. So we know this is only going to undergo meiosis. It undergoes meiosis. And we know what is the result of meiosis? 4 daughter cells. So the 4 daughters are called the 4 megaspores. So your next diagram shows the 4 megaspores. 1, 2, 3 and 4. 4 megaspores that have developed from the megaspore mother cell. Now of these 4 megaspores, the first 3 degenerate. So I am just striking off the first 3. 1, 2 and 3. The first 3 degenerates and the 4th megaspore becomes the embryo sac. So among the 4 megaspores, 3 degenerates and only one megaspore becomes a functional embryo sac. Such a development of the embryo is called monosporic development. So what is monosporic development? Of the 4 megaspores produced, 3 degenerates and only one becomes the embryo sac. Such a development is called monosporic development. Now we have the next diagram with an embryo sac and a single nucleus. It has a single nucleus. Now what happens to the nucleus? This nucleus undergoes mitotic division. And in the beginning of the class I told you the difference between meiosis and mitosis. As a result of mitosis, 2 daughter cells are produced. So the first mitotic division, 2 daughter cells are produced. Then second mitotic division, again 2 daughter cells are produced. And third mitotic division, each of them produces 2 cells. So totally how many cells are produced? 8. So this is how this single nucleus become 8 nucleated. So we have an embryo sac with a single nucleus. In the very next diagram we have an embryo sac with 8 nuclei, and look at the arrangement of the nuclei. They are randomly scattered. They are arranged here and there in the embryo sac. Next what happens is, in this diagram itself I'll depict orientation of these 8 nuclei. 3 nuclei goes to the micropylar end. 1, 2, 3 goes to the micropylar end. But before going into the orientation, like how in the social studies you have map activity, you have the North Pole and South Pole. Here I'm going to indicate 2 poles. This pole is called the micropylar end. And the upper pole is known as the chelazal end. So with respect to these ends, you're going to say the orientation. So of the 8 nuclei, 3 moves to the micropylar end, 3 moves to the chelazal end, and 2 remains at the center. Once again, the 8 nuclei gets oriented. 3 moves to the micropylar end, 3 moves to the chelazal end, and 2 remains at the center. Now, the last event is the layment of the cell wall. Cell walls are laid around the nuclei. At the micropylar end, 3 separate cell walls are laid surrounding each nuclei. See? And this structure constitutes the egg apparatus. Once again, I repeat, cell wall is laid. At the micropylar end, 3 separate cell walls are laid around the 3 nuclei to form the egg apparatus. At the chelazal end, again 3 separate cell walls are laid. 1, 2, 3, and these 3 cells constitute the antipodals. And in the center, there are the 2 polar nuclei. It is surrounded by a common cell wall, unlike the others. It's surrounded by the common cell wall, and this is called central cell. So we have the final diagram displayed here. We have the antipodals. At which end? At the chelazal end. We have the egg apparatus at the micropylar end, and this is called the central cell, which comprises the 2 polar nuclei. Thus, we can describe the typical ovule as 7-celled 8-nucleate structure. Why it is called 7-celled? Because there are 3 antipodals at the chelazal end, 3 cells forming the egg apparatus, and a single cell at the center which forms the central cell. But the nuclei number is 8. So once again, a quick recap of megasporogenesis. What is megasporogenesis? It is a development of megaspores from the megaspore mother cells by meiosis. Your second point, a brief description of the carpel. The carpel comprises the stigma, style, and ovary. And within the ovary, the ovule is located. The ovule is connected by a stalk called the funicle. Now, third point is the structure of an immature ovule. An immature ovule is surrounded by 1 or 2 protective layers called integuments. This integument encloses a mass of cells called nucellus, and one of the nucellar cells, which is large, containing dense cytoplasm and prominent nucleus, serves as the megaspore mother cell. Next, the megaspore mother cells undergoes meiosis to form the 4 megaspores, and they are arranged in a linear fashion, 1, 2, 3, 4. Of these 4 megaspores, 3 degenerates. The first, second, and third degenerate, and only the 4th megaspores become a functional embryo sac. So we have in the next diagram, ovule with an embryo sac comprising 1 nucleus. Now, this development of the embryo sac from 1 megaspore is known as monosporic development. Next, what happens to the nucleus? The nucleus undergoes 3 consecutive mitotic divisions to form 8 nuclei. So we have the next diagram with 8 nuclei randomly arranged. Let's count. 1, 2, 3, 4, 5, 6, 7, 8. They are randomly arranged. And the penultimate event is these nuclei gets oriented. 3 moves to the chelazal end, 3 moves to the micropylar end, and 2 remains at the center. And the last event is the cell wall layment. Cell walls are laid separately around the nuclei at the chelazal end to form the antipodals. Again, cell walls are laid separately around the nuclei at the micropylar end to form the egg apparatus, but a common cell wall is laid around the two polar nuclei at the center to form the central cell. Thus, I conclude that the typical angiosperm ovule is 7-celled, 8-nucleate stage. I hope you have understood the slot. The next slot I'll be dealing with microsporogenesis. Thank you.